So first of all, etylation is how a plant adapts to growing in the dark. The 2 times that I germinated the seeds, they were placed in a dark cabinet. The seeds' phytochromes were not absorbing any kind of light for the first 2 days of development, so the phytochromes were in their PR or inactive state. And so as you can see in these first 2 squash seed pictures, the stems are like white and there's no green on them, so they were fresh out the closet. And then here are the mung bean seeds that I germinated like fresh out the closet, so they didn't have anything. So how seeds actually break through their shell to like sprout is with the hormone germinans, which basically what happens is water enters the seed. In this case, it was through the paper towels I used to germinate them, and the embryo produces the hormone so that the seed can break through the endosperm, which provides sugar for the seed to sprout. And you could see that in these next couple pictures where it's like the seed still isn't like fully out yet, but like you could see the little sprout each day getting a little bigger. De-etylization is the process that a plant goes through once it's exposed to light. And as you can see in these next couple squash seed pictures, that's the process that it's starting to go through. And basically what happens is the phytochrome responds to the light stimulants and it goes into its PFR or active state. And that's a state of cellular response, which basically helps the seed germinate and continue to grow stem and roots. And in the squash seed, you can't see it as well because I messed it up in the way I placed it, but you can kind of see the root coming out and the stem trying to leave the seed shell. So where I messed up with the squash seed was with its gravitropism, which is basically the process of how the plant responds to gravity. And what happens is the roots are supposed to grow down and the stem is supposed to grow up. But the way I had placed the seed, it was that it was on its side and it wasn't growing the right way. And it got to a point where the root came out and the root and the stems were present and they came out, but they just weren't going anywhere because there wasn't anywhere else to go because I didn't place it the right way. What I had tried to do was place the seed upside down, so maybe I could like restore the gravitropism and make it go the right way. But after a couple of days, I realized that it wasn't working and the seed still wasn't growing, so I decided to replace it with the mung bean seed that did end up growing. So I did add back in this picture of the ungerminated mung bean seeds because I want to talk about abstic acid, which is basically the hormone that inhibits germination. And this first picture of the mung bean seed is the only seed that germinated in all the seeds I put in the bag. So it's just that the other seeds had a higher amount of abstic acid, which means that they didn't germinate versus the seed that did. This new seed is also demonstrating hydrotropism because the seed is growing towards the water basin at the bottom. So next I want to talk about cytokines, which are named after cytokinesis, and they're produced in the actively growing regions of the plant, and they just grow. They basically make the plant grow. And as you could see in these pictures, the plant is growing like roots and a stem. So while the plant really isn't growing that much right now, I'm going to talk about circadian rhythms and how they've helped my plant. And they do, because it's basically how the plant knows what season of the year it is based on how much night and how much day there is. So as the days go on, there's been more and more light because as like the days get longer towards the summer. And this helps the plant because it knows how much like stuff it has to produce, like hormones and whatever, to make it help it grow. So it knowing what season it is and how much night and day there is, it helps it grow. So now my plant is getting to a point where it's starting to demonstrate the phototropism, which is how the plant reacts to light. And I added this picture from later on about how the plant is bending towards the window, which is the source of light, to just show how, like, which way the plant is bending. So the plant is able to like bend like that towards the light because of a hormone called auxin. And auxin activates the protein pumps, which produce more H+ ions, which make the cell membrane more acidic. And this change in acidity activates enzymes called expansins, which break the links between fibers of the cell wall, and that allows more ions in the cell, and then more water as well. And then this increased pressure, like by more water, causes the elongation of the cell, and it's able to bend like that because it responds to the light. So as the plant continued to grow, I noticed kind of the triple response of ethylene present, and I guess the stress that the plant is under, that it was kind of running out of space to grow in the little thing, because I noticed that there was a slowing of the elongation of the stem, and the stem was getting thicker, and that it was kind of curving. So I saw maybe that it was under a little bit of stress, but I guess there could have been more stress. So another thing I like to talk about is how I saw evidence of thigmotropism in the plant, because as the days went on, like there were more roots growing, and it kind of like pushed against the cardboard thing. And it made it so it was like bent back a little, and that was the roots were responding to touch. which is the definition of thigmotropism. So finally, I'd like to talk about if the plant was growing really tall, about how it would have more PR than PFR in its phytochrome, and this more PR means that it's growing really tall, so look for more of that far red light. So if it gets taller, then maybe it could find it. So yeah, that's pretty much it. That's all I need to talk about. I haven't really put this plant into a pot because of the whole corona thing, but hopefully when I get a chance to, I will put it in a pot. But yeah, that's it.